In this lecture, we will meet the course objective by understanding how the spread of communicable diseases can be controlled and prevented by current public health practices, policies, and regulations. This is by learning about immunization guides and programs in Canada and the vaccines that are routinely administered or recommended. We will also recognize relevant laboratory tests and terms which may be encountered in reports or in the literature. This is quite a long list of specific learning objectives for this lecture, including firstly, gaining an awareness of immunization guides and programs in Canada. Secondly, identifying different types of vaccines. And thirdly, recognizing different vaccine formulations. In the second part of this lecture, we'll understand how immunoassays are carried out and why they're used in the laboratory diagnosis of communicable diseases. We will also be able to understand how and why nucleic acid tests are used, as well as understand how viruses are detected by various diagnostic tests. The name for vaccines was derived from Edward Jenner's first immunization against smallpox, which used the vaccinia virus. This is a virus that causes cowpox, and vacca is the Latin word for cow. Vaccines use the specific memory of adaptive immunity to provide maximum and long-lasting immune protection from pathogens. Vaccines therefore prevent full-scale infection and disease when secondary exposure to the pathogen occurs. In Canada, the group that develops immunization practice guidelines for both public and private vaccine delivery is called the National Advisory Committee on Immunization, NACI for short. This committee also defines the most desirable immunization practices for healthcare providers to assess current recommendations and identify areas of excellence as well as deficiency. I've added some links below with more information on this committee, as well as a link to the Canadian Immunization Guide, which we will use later on in this lecture, as well as in other lectures. There are many types of vaccines that have been approved for use in Canada. I've provided a list here. The four major types are live attenuated vaccines, killed, intact, or disrupted vaccines, a variety of subunit vaccines, and combination vaccines. I will go through each different type in further detail in the next few slides, especially for the various subunit vaccines. Live attenuated or weakened vaccines are the only type of vaccines that infect the recipient. However, they cause infections that don't result in disease. They're less virulent strains of the pathogen and are usually produced after many generations of culture in the lab. They stimulate good protective immune responses with lasting memory, so that booster doses are usually not required for several years after the administration of the vaccine. However, these vaccines need to be monitored carefully, as there is a risk that they may revert back to a virulent form capable of causing disease. Live attenuated vaccines are not recommended for immunocompromised individuals or during pregnancy. Examples of live vaccines are the measles vaccine in combination with the mumps, rubella, and chickenpox vaccines, all of which are live attenuated vaccines. Due to the potential of live vaccines to revert back to a virulent state and the possibility of causing disease, killed vaccines have been developed as safer alternatives. Killed intact vaccines are entire or whole microbes that have been killed either by heat or chemicals, such as formaldehyde. Even though they are unable to infect, they still are able to induce immune responses. They're therefore safer than attenuated vaccines, but they don't provide protective immunity with a single dose, and multiple booster doses are usually required. Examples of killed vaccines are the polio vaccine, which is used in Canada. This has been inactivated with formalin, and also the flu vaccine, which has been disrupted using detergents. Subunit vaccines can either be purified proteins or polysaccharides. The pertussis vaccine is an example of a subunit vaccine that is made up of purified protein antigens of the Bordetella pertussis bacteria. The invasive pneumococcal vaccine is made up of the capsular polysaccharides purified from various strains of Streptococcus pneumoniae. Other subunit vaccines include inactivated toxins and recombinant vaccines. The diphtheria and tetanus vaccines are both toxoids, which are toxins which have been detoxified by inactivation with formaldehyde. The hepatitis B vaccine is one of the recombinant vaccines made by purifying the recombinant hepatitis B surface antigen produced from genetically engineered yeast. The COVID vaccines that most of us have received are the latest in genetically engineered vaccines. The more traditional ones are the AstraZeneca or Johnson & Johnson vaccines, which have the SARS coronavirus 2 spike protein DNA inserted into an adenovirus vector. These are harmless viruses that are unable to replicate. Instead, they are used as delivery systems that enable the spike protein gene to enter host cells. Once inside, the host cell produces the SARS coronavirus 2 spike protein. The mRNA vaccines also use the same principle, except that messenger RNA is used instead of DNA. RNA is very unstable and requires storage at very low temperatures to prevent degradation. This is why the Pfizer vaccine requires storage in ultra-low freezers. The Moderna technology, which is proprietary, enables the vaccine to be stored at a higher temperature, instead in normal freezers. Nucleotide sequences can be changed, allowing the vaccine to target new mutant variants. There are several different types of vaccine formulations. The first type is the combination vaccine. This is a type of vaccine which provides protection against several different diseases, so that adherence to recommended immunization schedules can be facilitated. It also potentially reduces the amount of adjuvants and preservatives that would have been received if the vaccines were administered separately. Examples of combination vaccines are the diphtheria, tetanus, pertussis, polio, and Haemophilus influenza type B vaccine, also called the 5-in-1 vaccine or DTaP IPV Hib vaccine. Another combination vaccine is the MMRV vaccine, which contains the attenuated measles, mumps, rubella, and chickenpox viruses. Polyvalent vaccines are a little different to combination vaccines. Instead of providing protection to a variety of diseases, polyvalent vaccines only protect against one disease. They contain several different antigenic variants to widen the range of their protective efficacy. An example of a polyvalent vaccine is the seasonal flu vaccine, which usually includes 2 strains of influenza A and 2 strains of influenza B. The virus strains are reviewed every year, and the strains chosen for inclusion are the ones that are circulating predominantly based on surveillance data. Conjugate vaccines are rather different to combination or polyvalent vaccines. These vaccines usually have a subunit vaccine, such as a purified protein or polysaccharide, conjugated to another protein, such as the tetanus or diphtheria toxoid. An example is the Haemophilus influenza type B vaccine. The capsular polysaccharide is covalently bound to tetanus toxoid. All the vaccines we've covered so far are used in a process called active immunization, as it involves the use of antigens or toxoids. Another immunization process is called passive immunization. In this type of immunization, antibodies or sera are used instead of antigens and toxoids. Passive immunization is used when vaccines for active immunization are not available or are contraindicated. This type of immunization could be used to provide protection after exposure to the pathogen, especially if the individual who has been exposed has not been previously immunized. In this case, it is called post-exposure prophylaxis. Passive immunization is also a good alternative for immunosuppressed individuals who are unable to respond well to active immunization using vaccines. Unfortunately, passive immunization does not provide a long or complete protection, but is a good alternative to active immunization in the scenarios described on the previous slide. The antibodies are usually obtained from pooled human plasma, which contains mainly IgG with small amounts of IgA and IgM. In order to prevent any transmission of blood-borne pathogens, donors are screened for previous exposure to communicable diseases, and any pathogens that may be present are inactivated by heat treating the plasma or using other methods of viral removal. Animals are also good sources of hyperimmune serum, especially large mammals such as horses when human products are not available. However, using serum from other animals may cause serum sickness when the human immune response reacts to non-human antisera. It is a type of hypersensitivity which may manifest as fever, rash, and joint pain. Now that we've learned about different types of vaccines and immunization procedures, let's return to the first learning objective of this lecture, and that is to gain an awareness of immunization guides and programs in Canada. The first of these is childhood immunizations, which are routine. This includes the combination vaccine for diphtheria, tetanus, pertussis, polio, and Haemophilus influenza type B, which is administered at 2, 4, and 6 months of age, together with the polyvalent pneumococcal vaccine. At 12 months of age, a boosted dose of the pneumococcal vaccine is required, together with the meningococcal C vaccine, the measles, mumps, rubella, and chickenpox combination vaccine, and the flu shot. At 18 months of age, boosted doses of 2 combination vaccines are required. The first is the diphtheria, tetanus, pertussis, polio, and Haemophilus influenza type B combination vaccine, and the second, the measles, mumps, rubella, and chickenpox combination vaccine. If the 2 combination vaccines are not given at 18 months, they need to be administered between 4 to 6 years of age, that is before the child attends grade 1. This means that before any child can attend elementary school in Canada, they should be immunized against 12 different diseases. Once in school, a booster shot of the meningococcal C vaccine should be given between 9 and 12 years of age, together with the recombinant hepatitis B and the recombinant polyvalent human papillomavirus vaccines. Before leaving high school, the combination vaccine for tetanus, diphtheria, and pertussis is administered. But this is a different vaccine to the one used for childhood immunizations, as it has lower doses of the diphtheria toxoid and pertussis antigens to reduce any likelihood of adverse reactions. After school, young adults are recommended to have the tetanus and diphtheria combination vaccine every 10 years. The pertussis vaccine is recommended for adults who have not been immunized before leaving high school, especially those who are working with young infants. Both pertussis and polio vaccines are usually administered in combination with the diphtheria and tetanus vaccines, but it's the one with the reduced diphtheria and pertussis toxoids. Similarly, the meningococcal C vaccine is recommended for any young adults who have not received their booster shots in school. There are quite a number of vaccines recommended for older adults, and this varies with age and previous exposures. The measles and mumps vaccines are only recommended for those born in or after 1970, based on the assumption that anyone born before would have been infected and would have natural immunity. The rubella vaccine should not be given to women during pregnancy, but after delivery. The human papillomavirus vaccine is recommended for both women and men who are younger than 27 years of age, while the chickenpox vaccine for adults who have never had chickenpox or antibodies to the varicella virus and are between 18 and less than 50 years of age. The vaccine for shingles, which is caused by the same virus that causes chickenpox, is recommended for those who are 50 years or older. For seniors, the pneumococcal vaccine is recommended for anyone who is 65 years or older, as well as the flu vaccine, which is actually recommended for all age groups. However, a higher dose flu vaccine is recommended for those who are 65 years and older, as it provides better protection in the elderly. The 3 general categories of recommended immunization for travelers. The first category includes the vaccines that are part of the primary series of routine immunizations or their boosted doses. The second includes vaccines that are required by international law, such as the polyvalent meningococcal vaccine for Saudi Arabia and yellow fever vaccines that are needed for travel to South America and certain countries in Africa. And the last category includes the recommended vaccines to a variety of diseases. In the last category, the immunization schedule has to be customized for travelers and varies according to several factors which are listed on the slide. It's not just personal variables such as age, immunization history, and existing medical conditions, but the recommended vaccines are also dependent on the prevalence of the vaccine-preventable diseases in the countries that will be visited, and the duration and the nature of travel. This is because travelers visiting more remote and rural areas may experience higher transmission of certain diseases than those staying mainly in urban hotels. Travelers, therefore, need to visit a travel clinic well in advance, usually a month or more, to ensure that they have enough time to develop protective immune responses after immunization. There are several recommended vaccines for travelers, and a number of diseases are listed here on this slide. I won't read them out. I would just like to comment that these vaccines are not normally covered by the government, and you'll need to pay for them out of pocket. And some of them can be quite expensive, so they do add to travel expenses.